Iowa. It's good. All right, and this is the index map from my PhD dissertation. Unfortunately, it was done in the 70s before we had really high resolution graphics. But I think you get the idea. Basically, all the dots on there are data points that I had. Some of it was from USGS reports that I got faunal information. Some of it I went out in the field in 1979 and 80. And I think it was 79, I worked mostly in Wyoming. In 80, I worked with up in Montana. And I got data on 183 species of mollusks. Most of them were bivalves. All right. And back then it was township and range to figure out where you were. You see there's some red dots there. Those were the main samples that I used that were collected. All right, so let's take a look at some of the more common fossils of the Cretaceous seaway. When you think late Cretaceous, you think of bivalves, and in particular, inoceramid clams. They were the major fossil that I worked with during my master's and PhD work. Also had snails and cephalopods, in particular, these weird heteromorph Scaphites things that were uncoiling. If we look at the chronostratigraphy and some of the biostratigraphic zones, I worked mostly in the Upper Cretaceous. That would be lower to middle Coniacian. And here you see some of the main biostratigraphic zones that I dealt with. Earl Kaufman in particular defined many of these or set up many of these biozones. And one of the things that I found when I was out in the field, they were really easy to identify. It was easy for me to tell where I was in the biostratigraphic zones because these inoceramids are very distinctive. So you start on the bottom here with these Mytoloides inoceramids. And as you go up the section, you can see these things getting bigger, more coarse ornamentation in them. So this one up here is a Cremniceramus deformis, which would tie in here. And then when you get into the middle coniation, you get these Cremniceramus browni and Vulviceramus grandis. These things get to the size of basketballs. So there's a gradual change as you go up in terms of the size and the shape of these inoceramid bivalves. So I started with my master's thesis and focused mostly on Wyoming for that. And this diagram shows the major formations that you see in Wyoming. So you have the Frontier Formation over here. The Cody Shale is in here. And then the Niobrara chalks and limestones go in there. So these are the three main formations that I worked in for my master's and PhD work. And the western part of the seaway have nice cyclical deposits. You go from from shales to coals to sandstones, and it's sort of a repeating pattern that we see here. And as part of my master's and dissertation, I wanted to figure out what the environments were like. This was a paleoecologic study, so that was important. Here you see a picture that was taken of me by one of my field partners in the Frontier Formation. So you get these really outstanding sandstone formations of the frontier. And in the background, you get to see the Cody Shale. Have any of you worked up in Wyoming or Montana in these areas? No? Okay. Too far south. Okay, got it. All right, so there was a geologist, now USGS, Ryer, who came up with a couple of models, okay, for these cyclical deposits. And there were two possibilities that I looked at. One is a delta model where these represent the sandstones, and crossbed sandstones represent channel bars. Then you go into the shales, which are levee deposits, and then the marsh would be the coals. Or the other model is a barrier island model, where you start with a barrier island like you see off the coast of New York today, followed by a lagoon, and then you get a marsh above that. So there were two sort of models that I looked at for my dissertation, and eventually ended up sort of merging them together because I thought that there was evidence for both. So here you see a reconstruction of what Wyoming may have looked like during the Middle Coniation. So basically what I did was I had all the data on these bivalve or these mollusks, and I put them into a large database. And what I did was I ran a cluster analysis. I'm not going to go into a lot of detail about this, but basically what the cluster analysis does is it looks at similarity between the samples, and it groups them together. So you see some crosshatch marks here, okay, showing that the analysis is pretty much put these samples together and put these together. I sort of looked for natural breaks in the clusters, okay, to distinguish them, and ended up with about 4 major communities of mollusks that existed during this time. And then what I did was I plotted up the location of these 4 clusters on the paleoenvironmental model that I came up with. So the 4 main communities that we have here are a cardiid-rich community near the Barrier Islands. Then there's these Metalloides communities on the Delta platform, and as you get further out, you get into more Inoceramid communities. And what I thought would be helpful here, rather than just trying to put up the data, is make reconstructions of what these things look like. So here's the pluricardi community, where you see these cardiid bivalves, and you also get scallop sort of organisms. Here's my attempt at sort of artwork showing these. So like I said, you get a lot of infaunal bivalves, very similar to what you see in modern environments today. And you get some snails running around here. You get some really deep animals like telinids here, and then penis coming up. You also get some reclining organisms like Gryphea in this community. Going offshore, the next one is the Metalloides paleo community. And now you see more of these weird cephalopods, Scaphites, swimming around, and then you get more recliners. You get bissally attached bivalves, and you still get infaunal bivalves here in this community. The next one is a Cremdoceramus or Inoceramous rich community, mostly infaunal organisms in this community, although you also get some epibiont oyster sort of things on the Inoceramids. And then furthest offshore, you get what are called these pycnodontid paleo communities, rich in oysters encrusting these bivalves here. So you get a gradual change or shift as you go offshore in these paleo communities. All right, like I said, one of the things I was interested in was using the data that I collected in my master's and PhD dissertation. And in particular, what I was interested in was, could fossils be used for controlling the location of plates in the past? If you're familiar with recreating plate distributions, you start off with paleomagnetic data, and you carefully measure the location or the orientation of magnetic particles in rock. And we have equipment that's capable of doing it. And basically what you can do is you look at the orientation, the deflection from horizontal, and that could give you an idea of the latitude that the rock formed in. You can also see the rotation this way, so you know how much the continents or the plates have shifted. The problem is that there's a symmetrical magnetic field, around the Earth. And because of that, we can't get paleolongitudinal data. It's sort of been a bugaboo among geologists and really limited our ability to create reconstructions of what the plates were like in the past. So here was my idea. I decided to, one year, get lots of data on modern bivalves. I collected almost 1,000 species of data. And all I did was I went in field guides, and I created like all these provinces here and just made a note of what species are found in each province. And I put that in an Excel spreadsheet. And this shows some of the data that I was able to collect in sort of a Venn diagram. So we have up here a comparison between species of clams that are found in Western Europe versus those in South Atlantic. So I'm comparing European with American faunas. Then here shows West Indies versus Panama. So this would be the Atlantic versus Pacific. And then this would be North Atlantic versus South Atlantic, comparing to see if these clams were the same. And this paper was published in 2008. And basically, I was looking at barriers, how effective they are. And statistically, what it turns out is comparing West Europe with South Atlantic, the Atlantic Ocean is a very effective barrier controlling the distribution of clams. Comparing the West Indies with Panama, Pacific versus Atlantic, you got the Panamanian Bridge. It's an effective barrier. What's interesting is if you compare North and South Atlantic faunas, they're the same. They're basically a result of random mixing. So there's no significant difference, okay, between those faunas. What I did next was I calculated the similarity between provinces, and I used what's called a Jacquard similarity index. So I looked at the number of species in common and divided it by the diversity of the two. And there are a number of different ways of doing this, but I found that Jacquard was the best. It included both changes in the composition of faunas between two areas, as well as the diversity of each. And it ranges from 0 to 1. So 0 means faunas are totally different, and 1 means that the faunas are totally the same. And I ended up with a mathematical law. You know, I love throwing this out. I taught high school and college for 33 years in earth science and about 18 years in college. And, you know, if you go through science classes, you go into chemistry and you go into physics, there's a lot of mathematical formulas. You know, when I had physics, we learned F equals ma, force equal to mass times acceleration, right? When I took chemistry, I learned the gas laws. You know, PV equals nRT. Remember that? Sitting through those classes. I remember when I was at the University of Chicago, Tom Schaff, one of my evaluators, came into the lunchroom and turned to the students and yelled, "How come there's no gas laws in paleontology?" Okay, it was a comment that stuck in my mind. By the way, Tom passed away, unfortunately, but his comments stayed with me as I was doing this research. And I said, there should be. It's just a matter of looking for them. Use the same sort of methodology as physicists use and see if we can come up with some kind of mathematical relationships. And Dr. Schaff was absolutely correct. There are mathematical laws that relate faunal similarity to distance. It sort of makes sense, right? If I've got two areas here and they're close together where waves and currents can mix the larvae, they're going to be the same, right? And if I separate them, okay, what's going to happen is the larvae are going to eventually settle out. So the species are going to be different. So it's common sense that they're going to be different. In fact, it turns out that the relationship between how similar two faunas are versus distance is very similar to radioactive decay. It follows a logarithmic decay sort of formula. And so I was able to come out with this and publish this in 2008, and then also refined it later that we'll get to. So what does all this mean? What it means is that now I have a formula relating how similar two faunas are versus distance. And what I can do is invert it. I can get data in the past for the Cretaceous, for Wyoming and Europe, and I can calculate what the distance should be between them. And what I ended up with was an estimate of about 6,600 kilometers. I remember getting this result in 2008, and I'm going, oh my gosh, if you look at the separation from magnetic seafloor anomaly, you get 6,500 kilometers. And I'm going, wahoo, it works. Okay, I'm actually getting data that's similar to what you would expect from the oceanographic data. So the next thing was to see if I could fly without a safety net. What if you don't have the magnetic seafloor anomaly data? Could you use fossils to move continents and reconstruct where they were in the past? So that's what I gave a shot. And I was on a field trip in Virginia, and I just hit one spot. There were tons of clams all over the place. So I'm going, gee, there must be a lot of data out there. How many of you know about the paleobiology database online? Good, all of you should. It's an amazing source of data, of taxonomic information as well as location. It's easy to get access to. You don't have to sign up and create an account or anything. So I went and I started to get data on bivalves for the middle order, for the late Ordovician, for a number of different plates. This time I used a slightly different faunal similarity measure, the DICE, but basically it's the same idea. It's just an equation that determines or is based on the number of species in common versus their abundance. And again, I got a nice relationship between how similar two areas were versus distance. So now I've got a way of calculating the distance in the past based on fossil data. And here you see the diversity for Baltica, South America, England, Scotland, Ireland, the DICE indices, and from my formulas, what the distance should have been between those two areas. And what I decided to do, this is, by the way, this is relative. Okay, it's not absolute distances. Okay, what I decided to do was to make Avalonia, England, my prime meridian for 500 million years ago. And then I just measured everything off, okay, from Avalonia. And we get a reconstruction that looks like this, showing where the plates were. Whoops, let me go back here. Okay, it shows, that's what I want. It shows the location of these plates relative to England and Wales. For Baltica, this would be like the Scandinavian countries, Sweden, Denmark, and so on. Here is Laurentia, and put South America here. And it agrees pretty closely with reconstructions on other organisms. So I thought that was pretty cool. All right, so anyway, I've thrown a lot at you. Okay, any questions about the work that I did at the Cretaceous or this plate tectonic information? No, pretty quiet.
Real quick.
We got a question in the back. Sure. What do you mean, the error bars? Yeah, actually, the first draft I submitted to paleobiology, I just read on a computer, and I had it out to like 6 decimal places. And one of the reviewer came back and he said, "You really think you can estimate the distance between plates to the centimeter?" And I thought, no, probably not. So I had to whack off some of the decimal places that were there. So I'm not sure how you would actually estimate the error bars that are involved in this. I'm thinking maybe 100 kilometers, something like that. I would say this, for the data that I had, it worked out pretty well. I did try the Devonian data set, and that didn't work out as well. So I'm not sure why. I talked to, how many of you know Chris Gotees? He was a graduate student, went to the University of Chicago. You don't know him, but you know his maps. Okay, they're used just pretty much ubiquitously. And I asked Chris about it. He worked with Fred to put them together. And basically he said, what you should do if you really want to continue this and improve the accuracy is go through and redo your analysis, but break it down into latitude versus longitudinal components. So if I was to redo this research, I think I would do that. And because there's clear latitudinal gradients, okay, due to temperature and so on. And there's also longitudinal gradients. James Valentine at the University of California showed that a lot of it's due to upwelling differences that control the faunas. And it would be interesting to split it into components, just like physicists would do. All right. Any other questions? No.
Online.
Yes.
Do you know if anyone's tried this with the terrestrial vertebrate record?
I have never done it with terrestrial.
Okay. Thank you.
I would suspect it would be, think about that a minute. Certainly it's different barriers that are involved. In the case of marine realms, you got to worry about deep oceans, separating out the faunas or land barriers. It's opposite. I was just attended by, there was a graduate student at Yale said the opposite occurs when you're dealing with terrestrial faunas. Like, for example, if you look at dinosaurs, in the early Cretaceous, you have things like Acrocanthosaurus that are running all over the place because the seaway isn't established. Then the seaway gets established in the late Cretaceous, and it forms a very effective barrier preventing dinosaurs in the west from going to the east. What fun question I like to ask is, why don't we have any Triceratops or T-Rex in areas like Virginia, New York, whatever? It's because the Cretaceous seaway prevented them from going. But I haven't thought about trying to see if if there's any relationship with terrestrial faunas. In the
Okay.
marine realm, you can have much larger distances, so it works a little bit better. But if you
Uh-huh.
got some data and want to try it, let me know.
Oh, I will send you an email then because I think it'd be really interesting to actually look at the Triassic using this kind of
Okay.
method with Pangea.
All right. Yeah. Like I said, I put my email up there. I'll share my personal email with you, Will. Please send me comments because I'm looking forward to what you have to say.
Wolf has a quick question.
Yes, sir.
Okay. Hi, Doug. It was nice talking with you earlier.
Can I be heard here?
Yes, Ken,
Oh, yeah.
you're going through the speakers.
I'm never quite sure if we're connecting or not. I'm really excited about the way you put that together. I mean, it's a really excellent summary, I think, of ways of looking at paleogeography. On the other hand, as somebody who's worked in the coastal environments where um, terrestrial fauna, dinosaurs, et cetera, are involved, there's a huge difference in the lateral continuity of offshore fauna, um, deep sea fauna, that kind of stuff, and coastal environments. And you may have everything, especially in the Middle Cretaceous, which is, I don't know, kind of extends up to the Campanian maybe or Coniacian, and the number of microenvironments that we might encounter. And those are usually not preserved very well. And so that's kind of the space that we've been working on because the dinosaurs are living on the coastline, um, in the swamps and forests and lakes and streams or whatever it is that feeds to the coastline. Um, I don't want to be too critical because all models, have a sample size or a grid size or whatever you want to say. But I think there's a lot more diversity in the coast that could be incorporated in kind of what you're doing. On the whole, I think you're making a really great argument for ways of looking at paleofauna. And it also kind of contradicts a little bit with, I have to say this as a disclosure. Roel Kaufman was my second major advisor, and I am very familiar with inoceramid biostratigraphy. Um, they still, in my opinion, I'd like to hear your opinion, are kind of enigmatic. Are they living on the deep sea floor in anoxic environments, um, chemosynthetically absorbing sulfur and other things? Because we find them in places that almost nothing else can live. And at the same time, we keep finding them in coastal environments. And I'm a little confused as to why that's the case. Are they that distributed? What makes them that specific? But I'd like to hear your opinion on that.
Yeah, you raised a couple of questions, so I think I need to break it down a little bit. And the first comment that you made about environments is a very good one. In fact, the first reviewer of the paleobiology paper, uh, it went. This is a great quick story. What happened is I submitted it to Peter Wagner at the Smithsonian, the first draft, and he said, "Gee, we don't have anybody here at the Smithsonian that's really good at this. I have a mystery reviewer, and I'm going to send it to him and see what he says." Okay, went to the mystery reviewer, comes back, and he goes, "It's not bad, but I think you need more data and you need more analysis." And he was saying specifically what you were saying. Is there a facies component here? Sands versus muds versus shells. So he said, "Can you break it down by facies?" And I did. And it turned out it really didn't matter a whole lot, okay, when I was looking at it. It took a year, resubmitted the manuscript, sent it to mystery reviewer. This time it came back, Peter, good at data, good analysis, publish it. Turned out it was David Lees at the Royal Museum of Natural History, one of my real heroes, so to speak. So, and then Peter eventually published it. So that was the first one. And the second question you had about inoceramids was about are they chemo symbiotic, Doug? Do that again.
Yeah,
What was she asking about?
well, they show up in black shales, right?
Okay.
At depths that, plankton can fall down to. And I did work on that, especially the black shales of the Western interior. But they seem to show up as articulated seashells in environments. They're pyrite, for people who don't know, they've been diagenetically altered to iron pyrite. They're beautiful, along with the ammonites. And so those are very reducing, very anoxic, very hostile environments that nonetheless grow a lot of bacteria and other things that, well, there's a critter for almost every environment, including your sewer, you know. So they, at the same time, keep showing up in places at, for instance, Chaco Canyon, where we have the dinosaur fossils that Brent and Adrian and Ben and I started working with years ago on. And there seem to be tidal channels that have Inoceremus at very, very shallow depths. There's a lot of different possible, I won't even go into that. But I was wondering if in all your work, if you've developed a uh, kind of ecotaxological or environmental, I don't know, model or even sensibility about where inos are most common.
Yeah, good question.
That was Earl's thing.
Oh, it was.
Earl was the guy, you know.
I know. Okay. Well, all right. Let me see if I can respond to Doug. First of all, inoceramids were cosmopolitan, and which is sort of ironic because they are, you know, you think of these things as big, heavy shells, but they.
I think you guys hit mute in the room because the audio just dipped.
Everything went dead.
Yeah, I think it looks like they just muted based on their profile. Oh, and I see people in the video running up. Thank you.
We scared them away, I guess.
No, I see Ben messing with it. It looks like I can't tell because the camera's far away.
Okay. Can you hear now?
Now we got you. Thanks.
Okay, so one, they're cosmopolitan, probably because of their larval distribution. The second thing I would say, I didn't include it. I came up with a whole taphonomic model for this, and basically, Robert Aller, he was an early diagenesis chemist, said to me, "Think about it this way: you have organic matter in these shells, okay, and most of them they're in muds, but they're in concretions. And as the organic matter is decomposing, what's happening is it's mixing with iron and sulfur, or it's producing sulfur, as I recall, by the breakdown of the organic matter. And you introduce iron, and boom, you get pyrite. So you're absolutely right. These are reducing conditions, but that's probably a taphonomic sort of thing rather than an environmental issue. And that's the quick answer I can give to it. And then, do we have another question?
Yes, we do. If back to this person, a very Sue's Liviat. I'm gonna ask them to unmute. You got a question? Your hand is up.
Hello, I was just gonna say, can you hear me?
Yes.
So I think last time you were saying essentially the reason why Tyrannosaurus and Triceratops are not found in Appalachia is because of the Western Interior Seaway. However, I think the Western Interior Seaway, it was still there, just North America and, well, Laramidia and Appalachia were connected. And also, the newly resurrected Nanotyrannus is potentially a relative of Dryptosaurus, which potentially originated from Appalachia. So still haven't found Tyrannosaurus and Triceratops in Appalachia or anything related to it.
Okay.
It's also the case that we do have a Tyrannosaur, Appalachiosaurus from the East Coast, and from other areas around the world. So that could use a little updating. How they got there, what causeways there may have been across the seaway at different times, is still a very interesting question, I think.
Okay.
You got another presentation?
Yeah, I don't know how long you want to do it. We're at 8 o'clock. This is.
Because I'd love to get to your.
Why don't we get to the trial by one? We'll skip the dinosaur one. You want to do that? No, everybody wants me to do the dinosaur one? File through. All right. I'm not going anywhere. You want to do dinosaurs? All right. All right. Got it.
Dinosaur. And then we got a screen share. Okay, Sherwin, can you see it on that screen?
All right.
Great.
I think we're good. All right. Boy, in New York, my talks went a lot faster. I didn't have all the technical problems. All right. So a number of you, I think it was announced during Aaron LeCount's lecture that I've been doing computer simulations, virtual reality simulations with dinosaurs. In 2022, I went out west. There we go. Went out west and I met the fine folks at Dinosaur Ridge. And I just finished a virtual reality simulation on the early Cretaceous. And I showed it to Jeff and Aaron, and they loved it. They said, "Oh, yeah, you got to do the Jurassic and the Cretaceous." So I put together a multiplayer VR simulation for the Molluscan Formation, the Jurassic, about 150 million years ago. And this is what it looked like. And the intent was to create kiosks at Dinosaur Ridge where guests could go around and see reconstructions of what the dinosaurs looked like. And the simulation had a time clicker at the top, so it limited, how much time, because we didn't want people spending an hour there and and blocking it for other people. And as you ran around, it recorded the number of dinosaurs that you were found. The other thing that was really cool about this is that it used remote procedural calls. So as I had a couple of students running around in the VR headsets and they were finding these dinosaurs, as one person on the team found it, the information got sent to the other teammate or vice versa. So it was a way of quickly having people find these animals. And every time they found one, there was a pop-up that would appear that would tell you information about each one. And I showed this off at Dinosaur Festival in 2023. And this is what it looked like. You can see a couple of students in the background there with the VR headsets on, and they could see each other in the program. And then you had a parent on another computer so they could see what the kids were doing. And what I found out, to my surprise, was that the VR simulations were really popular. When I set this up for the museum, I sort of expected like teenagers and adults to be wanting to do the VR, because at the time that was what Meta was recommending. They didn't want anybody under 13 using the VR headsets. And I worked with about 3 other people. And as the guests came up, we said, "Hey, this is not recommended for little kids." And the parents said the same thing. "We don't care, strap it on their head." Okay, I'm here to oblige. So it was popular. So the next thing I had was, but I wondered if they were learning anything. So I went back to my high school in good old Franklin County, Virginia, got permission to use the simulation with some of the students that were there. And what I did was I gave a pre-quiz to the students, then I put them inside the simulations, and then we did a post-quiz. And surprisingly, the scores didn't go up that much, only 10%. I guess they're not reading when they're in the simulation. But what I found was really cool. If I stood behind them, if I used it as a teaching tool, scores went up 32%. I said, that's not bad. So that was 23. Then in 24, I said, let's try the Cretaceous. And I got better models. Erin was awesome. She said, here are the animals you want to include in your simulation for the Cretaceous. And here we can get these models from like turbo squid and so on. So this is one of the reconstructions I came up with for the Cretaceous. And here you see one of my helpers. We had a child with a VR headset on, and he was running around looking for him. We ran into some technical problems. I couldn't do a multiplayer VR experience. I just did a single player one, but I still got some really good results. I found that test scores went up by about 32% using a virtual reality simulation. The real surprise was how young the children were that wanted to do the VR. The average age was 11. I had parents putting the headset on kids as young as 2. Now, come on. Do you think the kid is really getting anything? where was Jeff laughing? I think what they were doing was they just wanted to have a picture of the kid in the VR headset. All right, they want to do it, that's fine. All right, so I did the Jurassic, I did the Cretaceous, then I said, why not do the Triassic? So I started to put together a very simple Triassic simulation with a Coelophysis dinosaur, and then I put a Phytosaur skull in it. And I work with a family here in Arizona, and there's a 6-year-old who really loved dinosaurs. So I made it multiplayer so that he and I could be in New York and he could be in Arizona and run around and play with dinosaurs. And so here you see a screen capture of that. And the cool thing was that the child really got into it. I played once, and then a couple of months later, he goes, I think we were on Facebook instant messages, says, can we play the dinosaur game again? So he was really excited about this. It was lasting over months. I said, sure. So you got the software on your computer, and I went on, and we were able to play it together. He also was learning his alphabet. So I said so that when he typed commands on the keyboard, like hide, it would trigger a behavior in the dinosaur, and he could see the thing sort of stick its head in the sand or whatever. All right, after that, in 2024, I went to Connecticut to Dinosaur State Park and met the park ranger, Mike Ross, who gave me a tour of the exhibits there. And he told me that he really liked my simulations, and he wanted one for the Triassic in Connecticut. And these tracks have been found, and Connecticut, to their credit, covered up with a dome to preserve the individual tracks. I was using the Unity game engine, but he wanted Unreal. He had some concerns about licensing. I said, okay, that's not such a big deal. He had dinosaur footprints, and he wanted like a VR simulation of the dinosaurs running across the plain. So I'm still sort of working on that with some friends in Connecticut. But this is what I came up with. One of the things I found out was Unreal was really good at modeling the landscape and the environment. And so what I did was I put together a simple reconstruction, and our own Jenny Borst got together with her on, I think it was Discord, and she took a look at it, and she said, here's the animals you need to put in your simulation. And like Aaron, she said, here's where you can get them from. Here's how much they're going to cost, which just saved me a ton of time. So shout out to Jenny. Thank you very much for your help on that. And this is what the reconstruction looked like. That was the finished product. And the main thing that the Virginia Museum of Natural History and also the Dinosaur State Park wanted, was the landscape. They wanted to get the idea across that in the Triassic, this is now East Coast. I know it's going to be different from Arizona. As North America and Africa started to pull apart, you get these rift valleys that are forming. You form sort of horse and graben features, and that was really easy to model in Unreal. And then I put in some lakes, and here are some of the phytosaurs and coelophysis that are in there. So I got a really good graphic representation of what the Triassic was like. So Dinosaur Days 2025 rolls around. I went to Virginia, and showed it off. We had people from Virginia Tech show up, and they brought some of the dinosaur fossils that they have, including T-Rex teeth. And there's me with, this is Darian Bradley and Logan Howell. They helped me run the simulation. Unfortunately, we ran into problems with the VR, some technical problems just before we done, and I'm not sure exactly what happened, but we ended up just using regular PC computer simulations. But students still liked it, maybe not as much as the VR, but it was still interesting. We did a pre-quiz, then a simulation, then a post-quiz, and saw a really nice increase this time. I don't know what happened, but we got a 38% increase in the information that the museum wanted guests to know. So it was 51% when they came in the museum and 89% when they left, which is not bad. All right, one of the things too also that we tried was we asked some sort of loaded questions. I wanted to know, did guests think that dinosaurs and people coexisted? And what I got was that 80% of the guests showed up said, no, they didn't exist together. We had about 18% said they did coexist, And after doing the simulation, that number dropped to around 3%. So I think the simulation was helpful in clearing up some misconceptions about that. I had a student in Arizona work with a student in Vietnam, a sort of collaborative project. The students wanted to get together and create an app for Android phones with nonetheless dinosaurs. And here's what it looked like. This was sort of clever. We went into Unity and we were putting them all together, and we decided to play around with textures. And we got a really interesting result, a transparent texture with a dinosaur sort of looks like a ghost image. And that was purely by accident. I said, that's kind of cool. You could create sort of a modern simulation or landscape and then apply a transparent texture like the dinosaurs are ghosts running around there. Maybe that's an idea. Where's Benjamin? It would be something you might want to consider. So it runs on PCs, but eventually, like I said, we want to make it sort of like a platformer game. And we came up with a multiplayer version showing something like this on an Android phone. And here's the freebie. I threw this out during Aaron's talk. Dinosaur Ridge and I decided we wanted to have a longer version of the dinosaur simulation. So we came up with this late Cretaceous game that looks like this. And you go in and you get to drive around a truck and walk around and find dinosaur bones. And to pay homage to the USGS, there's even a mini map here. And the mini map is a geologic map showing the formations, the K is Cretaceous, and then I think PG is Paleogene. And then it actually shows where the dinosaur bones have been found. So as you're running around, it sort of helps you to find and locate the bones. And then after you do that, you can create an inventory of these bones. Then you take the bones and you put them on the skeleton. So if you want to know where dinosaur anatomy is like, you can do that. And there's 3 levels. There's easy, medium, and hard. So when you start with easy, as you drag the bones in just about anywhere on the skeleton, it'll click in. And then medium and hard makes it, you have to sort of get them closer to their actual locations. There's also an activity where students can drag the main dinosaur groups onto a phylogeny or family tree of dinosaurs. There's a restoration of what the Cretaceous may have looked like. We can go around and see not the skeleton of the bones, but the actual dinosaurs. There's a trip that you can take to a modern fossil prep lab. And what I did was I got permission to scan in 3D models of dinosaur bones at the Virginia Museum of Natural History. So as you're running around, you see here's a Triceratops horn. And as you get close enough, your arrow keys actually allow you to rotate around and see the 3D image or the 3D fossil. And then some other information there about the fossil. You can also do an activity where you can drag the dinosaurs onto the geologic timescale. Christy Deathridge said, Spielberg has really hurt us. Everybody thinks that all the dinosaurs lived during the exact same time, which is, of course, not true. All right, and if you want access to this dinosaur software, there is a QR code, so everybody's taking out their cell phones, and there's a bit.ly link there if you want to do it, but Aaron said you're probably better off with the QR code. So feel free to download it and give it a shot. It's a large file. You download it, you unzip it, and then you run the application file to it.
And I can send it.
Yeah, that would probably be better. But I appreciate people, zapping the QR code.
I want to get on the trial by our activity that we'll have here in the room. 1 or 2 questions.
Okay.
That's super quick. Did you know that they were being secretly educated when they were using the software?
You talking about when I did it at the museum or the high school? Yeah. What do you mean they were? What do you mean by recorded?
Oh no, did they?
We had permission from in my high school. I had permission. There was parent permission slips that had to be signed by the parents saying for the kid to be involved in it. And if they didn't bring him in, they didn't do the headset. At the museum, the parents were with them, so they were approving, this. And you saw images. I asked permission from the parents if I could take pictures, and they all said, of course, that's not a problem. So that was.
Can you bring up the QR code again? We've got some people online that wanted.
Sure.
We'll give us a 5-second count here and get on to our next activity.
There's a question over here.
Question? Last question.
I just want to tell you guys, I really love
I'm trying to get there.
Stand up. We barely see you.
Good.
So you're gonna take Dr. Schmachtenberg to the task here.
Okay, good.
Okay, let's go ahead and get on to the trilobites. Let's see what Calvin's got for you.
Okay.
This one.
Yep.
Got it over there, Sherman.
Yep.
All right. This is literally hot off the presses. What I'm telling you tonight, I just finished this research up a week ago, and it's really not even finished. It's preliminary research, but it's on this particular trilobite known as Triarthris. It's a genus probably you haven't heard by the name, but if you've seen trilobites in any textbook, probably you've seen Triarthris. These things are very common in New York, not so much around here. And what's incredible about them is the preservation. You not only get the entire exoskeleton with these guys, but you get pyritization of the soft parts. You get pyritization of the antenna, the appendages, and so on. My evaluator at Cornell, John Sisney, worked out all of the skeletal musculature and the respiratory organs of these guys. So they're really cool. There's one that's up here. This is a Triarthris that I collected when I was a teenager and really got excited about these guys. And then sort of I got involved with teaching and so on, decided to get back to it. All right, so it turns out that there are two main species of Triarthris, T. beckai and T. etoni. Beckai is the old one, and then E. etoni is the young one. And what I did was I put together some data. I'm still collecting it, and I'm really picky. I only selected so far about 96 images of these trilobites, although there are hundreds that are out there. And the reason will be an immediate apparent. So this lists the geologic age by biozones. These are graptolite biozones, from bottom to top here, oldest to youngest, superposition order. And the species are listed here. So this shows, the amount that I had for each of these. And this shows a complete Triarthris E. etoni from Rudimann's work in 1926. All right, what I was interested in was evolution, in this trilobite. And these things have been studied since 1838. And what I decided to do was do what was called landmark analysis. Mark Webster at the University of Chicago put together an article on how you do it. And it's actually pretty simple. What you do is you take images of these trilobites and you just click on the important points or landmarks on the trilobite. So here's the anterior part of the cephalon, anterior portion of the eye. 3 is the posterior position of the eye. 4 is the lateral part of the cephalon, so on, these glabella. And what it does is it superimposes an XY coordinate on each of these fossils and automatically records the coordinates for you. And then it does all the math. And what you can do or see here is this shows the location of these landmarks. I did about 8 of them here. I think it was about 8, and averages it out. So you can see how there were changes. So I looked at T. beckai from older horizons and younger ones. And I wanted to know if there was any stasis that was going on. Are they staying the same or are they changing? And the answer is they're changing. Even with a short stratigraphic interval, about 1 million years, you're seeing, for example, that the eye is moving a little bit towards the posterior of the shell. That's something that people have been arguing about in the literature is there stasis? And to me, I just haven't seen it in terms of my results. And it even gets more emphatic or obvious as you compare T. beckai and T. eatoni. And you see even a greater shift, okay, in the location of the eyes, okay, there. I like using vectors to show that. For some of you that are familiar with multivariate statistics, this is a PCA analysis. You take all the landmarks and put it into another program. It generates a PCA. And I know it's really hard to see this, so I'll break it down. What the PCA is showing is the older species. Whoops, I need to go back. The older species of Triarthropus are here on the right-hand side, and the youngest are on the left-hand side, and all these colors represent different species. So this analysis is able to split the Triarthropus by geologic age and also by species. That's statistically significant. And here's a phylogenetic analysis of that was produced by Ludvigsen and Tufnell. I think they're on the right track with beckai at the bottom producing eatoni and then some of these other species. And this is the latest thing that I came up with working with Chris McRobertson and Suni Cortland. What's going on with these trilobites? What's driving these evolutionary patterns that we see in the landmark analysis and the PCA? And I think the answer is sort of obvious. If you look at the eye, you can measure angles, okay, that are going on. So here's the eye. You can measure the field of view. So we know the trilobites use calcitic lenses in a hexagonal pattern and oriented so the main axis of the crystal was perpendicular to the surface. So we know roughly what the field of view was here, and it increased in general over time. Also, what I saw was what I call the azimuth angle. To simplify it, the eyes are rotating in a posterior direction by 10 degrees. And why would it do that? I mean, that's pretty cool, right? As I can tell you as a teacher, I wish that I could rotate my eyes in a posterior direction, see what's going on behind me. All right, but I don't have 7 million years like these guys did to pull that off. I think what's driving these evolutionary patterns is if you look here on the side, now this is a lateral view of Triarthropus that's pretty complete, but over here, it's been eroded by it's been eaten away by, I think, predators. And my number one suspect is orthocone cephalopods. There are like 40 species of these things going around. So what happens is these trilobites are in deep water, probably where it's pretty dark to start off with. And then what happens is the water gets shallower. And as it gets shallower, you start getting into the photic zone. And I think it makes it easier for predators to find these trilobites. And so there was an advantage, a natural selection, if you will, to rotate the eye, okay, in the posterior direction so you could increase the field of view and see these things, because the bite marks are not where the trilobites are able to see. And here's some data that I put together on these trilobite species. Over that. Okay, here's some possible examples of predation in some of these Triarthropus, where you can see, even if you have a spine here, that doesn't stop the cephalopods from eating on them. And here's another one showing predation in the pygidium. Here's some more parts of the pygidium rather than the cephalon. All right, so that's the results. I haven't published this yet. It's still in press. I hope to get more data on these Triarthropus trilobites. And what I really want to do, parents and teachers, I'd like to come up with a curriculum for teaching evolution with Triarthropus, because there's so many principles in evolution. I was telling Sherman and Benjamin, you know, these guys are great for looking at Cope's law. Did they get larger over time? How many species are there? Was there stasis going on, or were there punctuate equilibrium events? You can look at heterochrony, because Triarthropus, we've got the sort of the baby protaspid stage, the intermediate meraspid stage, and the whole aspids for beckai and eatoni. And so the end result is it looks like it's recapitulation. It looks like these things are starting with beckai in the juvenile stages of eatoni and then modifying it as they go through diatoni. That's interesting, because those of you that are familiar with heterochrony, the main driving mechanism, neoteny, it's supposed to go the opposite way, and these guys are breaking that sort of rule. And you can also look at allopatric speciation because the basin they're in starts to break apart as you get to the late Ordovician, and you can see species being derived. So there's tons of sort of lessons in evolution you can get from this Triarthris, and they're rather common fossils. The last time I was in central New York, I got about 5 or 6 of them within a half an hour. I know some spots you can get 700 of them, they've been reporting. So they're really easy to get. And for the activity, do you want to pass that out?
Sure.
Okay. I was at the American Museum of Natural History, and I was talking about landmark analysis with Scott Evans. And I said, you know, this isn't so hard. You know what it reminds me of? Dot-to-dot puzzles. Okay. You can take the results of a landmark analysis and make a dot-to-dot puzzle for actual fossil species and plot it for different species. And I got a feeling we're going to run out. So if you want this, email me and I'll get it to you because I'm not sure if we're going to have enough copies here. How are we doing, Sherman?
A lot of people know too.
That's what I'm thinking. All right. So maybe the thing to do is throw my website, my email up here.
So I'm going to go ahead and stop the recording. And I'm not sure if the folks online want to stay with us, but let's give Dr. S a hand.
It's been a pleasure being here. Thank you all for coming. Yes.
So, Dr. Describing what you're going to be learning there in terms of evolution.
You all know dots, right? I love these as a kid. You just take a pencil and you go from 1, 2, 3, and so on. I used to do it with cats and cars, whatever. This time, though, this dot-to-dot is for Rebecca, the older Triarthris. And if you do it here, this is for the younger one. And the goal is once you do the dot-to-dot puzzle, compare the two images. And you should see a shift in some of the features, specifically the eye, okay? And compare 3 and 4, okay, to the other landmarks in there. And you should be able to see what I was talking about on the slides. You should shift towards the back of the set line. This dot-to-dot is, I didn't make it up. All the dots on there were generated by the computer from live actual measurements of these trilobite species. So it's actual data that you don't know that you're actually analyzing fossil data with landmark analysis using a simple dot puzzle. And like I said, you'll share my email, right?
Yes.
All right, if you didn't get a copy of it, email me. I've got a digital, I'll send it to you, and you just print it out. I mean, if you wish.
I'm trying to see right. So you guys, I'm sorry, I was going to try and see if we could get the people online to actually see what was going on in the room, but my laptop is not making me see. Merlin, do you have your laptop?
Yeah.
You're on the.
I'm not. I got it.
Okay. All right. So for the folks on Zoom, we may go ahead and let you go for the evening. But again, thank you so much for joining us. I did record this, so I'm going to go ahead and put it up onto a Box account. Or actually, what I'll do is I'll put the link to it in Discord. And so what we're going to do is so with apologies to the folks on Zoom, we're going to go ahead and let you go. We're going to let Dr. S and Ben and some of the other researchers work with anybody that wants to try this exercise and learn a little bit about trilobite evolution. And we will see you guys next month for our next meeting.
Yep.
So, all right. Thank you, guys. We'll go ahead and let you go online.
Oh, okay. But if you have like the digital file, you can work that. Yeah. All right. To avoid.